Ferreira Ramos, coming from São Paulo also. He will talk about mathematical modeling of tumor progression on metastasis. Thank you.
Okay, thank you. So good afternoon. It's a pleasure to be here with you guys in Rio de Janeiro. So what I'm going to talk today is about the use of stochastic modeling to understand this phase of the tumor before invasion occurs. So when we are considering tumorigenesis process, there is, it may be divided in two main phases, a pre-invasive stage and an invasive stage. When we have an invasive stage, we may get to metastasis, but today I'm going to focus mostly at this time when we start getting the formation of a tumor. So it's a well-located region in a given tissue where you have abnormal cell proliferation, such that instead of keeping a balance on the cell population within the tissue where the amount of cells are tightly controlled by homeostatic mechanisms, you start getting abnormal proliferation and you get those tumors. Sometimes those tumors may keep growing. This is when it becomes an in situ at a pre-invasive stage, and then it goes towards different tissues through blood, and they become invasive and you get metastasis. So one of the conditions for this proliferation to start happening is when you lose a condition that is contact inhibition of proliferation. So if you consider normal cells, you take them, put them into a plate, to do some culture experiments. You will, and you watch them grow. And then at some point, you just decide to remove some cells and you keep watching to see what is going to happen. If you have normal cells, what is going to happen is that they will just cover that region, and they will stop proliferating. They will stop dividing, so everything is going to be okay. There won't be any abnormal proliferation. However, if you have cancer cells, instead of stopping proliferating, dividing when they cover the bottom of your well, they keep dividing and they start accumulating on the top of each other, and you get those tumor-like structures in wells instead of getting just a single layer of cells. This is contact inhibition is how they have named this phenomena here when you have the formation of a single layer, and whenever you have this accumulation of cells, then what they call that they lost the contact inhibition mechanism. So when you have this situation, they are considered as the analogous of the growth of abnormal growth of cell populations within a given tissue. Here we are considering breast cancer. So if we have a normal duct in the breast, there will be a single layer of cells here. However, if they lose some mechanism controlling proliferation, what may happen is that they start growing new layers here, new layers, and they will, here is at the moment when you have a carcinoma in situ, they will, until at some moment they disrupt this barrier here and start invading. By this time, what will happen is that this is a pictorial, just a cartoon, but one thing that is interesting is that this invasive time here just happens if the cells acquire a new phenotype. Okay? The cancer cells, if they don't change their phenotype towards an invasive phenotype such that they can colonize a different region of your body or of the body, they won't be capable of doing invasion successfully. Maybe they can escape, but they won't be invasive. The mechanism, the molecular mechanism underlying this abnormal cell proliferation and the formation of those tumor-like structures in a well, for example, is this membrane protein here called ecedrin. They bind to each other and they do some signaling that controls proliferation and or apoptosis and other mechanisms of for controlling cell population in a given region. So one question that we can say is, okay, so is this loss of contact inhibition an important mechanism underlying the capability of cancer cells to invade different tissue? So that is a natural question to ask. It seems to be so up to a certain level. Of course, I won't be crazy to say, oh yeah, it's indeed the only mechanism. That would be too much. But based on some experiments using a rat, that is the naked mole rat, it is an undergrad rat, it is a small one. They survive for something like 30 years, which is something that is very rare in nature for such a small animal. And particularly, we always associate cancer as a disease related with aging. So as older as we get, as higher our chances to get cancer. Those rats are living for 30 years, which in their scale, that is living like forever, and they have no cancer. The question is why? What is going on? What is the property that they have? So one of the mechanisms that they do have is that they have hypersensitivity to contact inhibition. So how did they measure this? Recently, Siluanov and collaborators, they made some experiments using culture experiments. So they just took cells from those naked mole rats, put into a dish, and let them grow, just grow. And then they took fibroblasts. And what they watched, and they kept watching how big would the cell population become within those wells. And what they measured is that if they take wild type naked mole rats and measure the populations, the cell population, the fibroblast populations, they would grow up to a certain limit and they would become stable. However, if they do a specific mutation at this gene, P16, what would happen is that the naked mole rats, the red curve here, cell population of fibroblasts would grow much higher. And they also did the same experiment using normal rats, mouse, with fibroblasts too. And they verified that the density of the cells within the well was, they were pretty much the same if we consider a mutated or a mouse. The experiment can be repeated using humans too. What's the difference at the genome level? The difference between the two species of rats is that the naked mole rat has this gene P16, which is not here for human or mouse fibroblasts. And this gene here, whenever it is there and operating normally, it controls cell proliferation such that it blocks it for much smaller cell densities in comparison with the situation when you don't have this P16 gene. So it is hypersensitive to existence of different cells around it. So this is a strong mechanism that controls cell population in a given tissue of naked mole rats. So it is a homeostatic mechanism. And see here that whenever we are talking about contact inhibition, we may be talking actually in terms of maximal cell density within a given well where we are doing our experiment. Okay, so what if, instead of having only a single cell in a given dish for doing the experiment, we use 2 cell types? Now, we can use, the red ones here are normal cells, keratinocytes, and we also put together melanoma cells. And we let them grow, put food and let them grow and try to see what is going to happen. What we have observed doing this experiment that is recently published is that you just take the well, put 9 keratinocytes per 1 melanoma cell, shake the well, put them all together, let them homogeneously distributed within the well, and wait. After a while, here, 8 days, what we are going to observe is that the well is going to be fully covered. But instead of getting a nice mixture of all cells, what we have is the formation of clusters of melanoma cells, those blue ones, surrounded by a sea of keratinocyte cells or normal cells, okay? Secondly, what we have observed is that the initial, the population of keratinocytes, it grows, as well as the melanoma cells, they too are going to grow, following a sigmoidal curve, like the logistic model, which is capable of fitting it. However, there is a new thing here. By the beginning of the experiment, we had a proportion of 10 normal cells per 1 tumor cell. And by the end of our experiment, we had a proportion of 4 tumor cells, 4 normal cells per 1 tumor cell. So proportionally, the tumor cells grew much faster than the normal cells, even though they were not the dominating cells within that region. So the question that we made was the following: okay, using logistic model is not enough for modeling this problem. What is the mechanism that is underlying this faster growth of the tumor cells, particularly because when we did the fitting of our data, we saw that the growth rate given by this parameter rho was pretty much the same for both cell types, either the cancer or the normal cells. So that was kind of let's say, a contradiction or unexpected. So what we did was, let's propose some type of agent-based model. So we considered a grid where each vertex of the grid we could or could not have a cell. The cells would go towards the following transitions. They could divide, they could they can or cannot be transformed from a normal towards a tumor cell. They will die spontaneously. They can quiesce, and they can migrate. Okay. But furthermore, we introduced a second property. When we were talking about contact inhibition, we are talking about densities. So whenever you have a loss of contact inhibition, you have higher densities. So in other words, when you have a higher density, what you have is that the cells are closer to each other. They are more packed. And when we have a strong contact inhibition, we have lower densities, so the cells are going to be far apart from each other. So what we did is the following. Let's consider that those cells are interacting as a hardcore potential, such that a cell has a smallest distance around it that it can permit the other cell to get. If we have normal cells, naturally, if we are considering an experiment with normal cells, their density are going to be smaller. So we can assume that they require exclusion diameter around it that is bigger than the tumor cells, which have higher densities, so they get more packed. So those shadowed areas are indicating those exclusion diameters. We are going to indicate the tumor cells by blue and the normal cells by red. The red area around a red cell indicates the exclusion diameter of a normal cell. On the other hand, those purple areas surrounding a red area indicates the forbidden zone for a cancer cell when it tries to approach a normal cell. So we are saying that normal cells establish a longer distance from, between them and the tumor cell. And we are assuming that the tumor cells have do not demand any exclusion diameter. They just accept any tumor cell to get as close as, as we can. We have, so in our grid, the distance between, the cells are going to be located at the vertex of the grid, and the distance between two vertices is going to be the minimal amount of edges uniting them. So the pattern that we obtain doing the simulation that I'm going to show the algorithm on the next slide is this one. And as you can see, it has a structure that is very similar to that one, this one that we showed in our experiment. We have a C formed by normal cells, the red ones, and we have some islands of tumor cells that are the blue ones. Okay, so how is our algorithm? What we do is, first we are going to, of course, because of the nature of our experimental arrangement, we assume that no normal cell is going to be transformed into a tumor cell because the time scale is not long enough for that, number one. And number two, the two cell lines are from different embryonic lineages, so there is no chance for a keratinocyte to be transformed into a melanoma. Okay. We are going to propose a discrete time Markov chain on admissible configurations. That is, cells cannot be closer than the exclusion diameter to each other, closer to each other than the exclusion diameter. We assume that each vertex of our grid assumes a state 0, 1, or 2. And so in order to compute the dynamics, what we do is the following. We take one of the vertex with a probability 1 over the total of vertices of our grid. So we take at random with uniform distribution one of our vertices, and we verify if this vertex is occupied or empty. If this vertex is occupied, it can remain occupied with a probability that here we assume that is proportional to the division, the probability, the division, cell division rate. We just took this parameter to be this value because we would like to reduce the amount of parameters of the model to as much as we could. And then it could also become empty. The vertex would become empty depending on two situations. First, the cell dies. The second, the cell migrates towards a different vertex of neighbor vertex of the grid. Okay, those are given in terms of this rates, the rate of diffusion and the death rate. And look that there is this number here that is the total number of empty vertex that can receive that cell, provided that if the cell gets there, it will not be closer than the exclusion diameter from any other cell around it. Okay. Otherwise, if the vertex that we take is empty, it can remain empty just because nothing happened in the grid, or it can receive a cell. This cell can be received because a cell division from other vertex or a cell migration from other vertex. And again, we do the same consideration that either the division or the migration depends on the total amount of cells surrounding that vertex and that can get there. Okay. And now we just run our simulations, and what we got doing this the simulation is that, okay, we start with an initial condition. We have 9 normal cells per tumor cell. We mix them and trying to get a configuration that is as more homogeneous as possible, and we wait our simulation to go. Okay, here we have iteration number at the time, so this is a grid that is 200 by 200. Parameter values are given here. Note that there is no cell transformation into from normal to tumor. So what we see is that we start getting some patterning, and by forming a sea of normal cells surround and small islands of tumor cells, and those small clusters start to get bigger and bigger, and at some point there is a transition here such that we go from a configuration where most of the cells are normal towards a situation where most of the cells are cancer, and at some point at steady state regime the domain is going to be fully occupied by tumor cells. If we take a look on the cell density within this spatial domain, what we get is that you see the normal cell density grows and reaches some plateau, and then the tumor cells also reaches a plateau, but you see that it keeps growing. And if we take a look on the ratio between tumor cells, normal cells over tumor cells, what we see is a similar pattern that we had observed experimentally. That is, the tumor cells, they grow, they start to prevailing within the spatial domain. Furthermore, what we see is that we have the formation of those clusters as well as we have seen in our experiments. So here is a sequence of images of that we got experimentally. So this is the first day, this is an intermediary day, 4th, and this is at the 80th day when we reached confluence. So here everything is well mixed. You see tumor cells, normal cells, and so on. Here we start seeing the formation of some clusters, but they are still sparse. And normal cells are also getting combined together. And then what we see is by the 80th day when we get a full confluence, or I mean, the whole dish of the experiment is fully occupied by our cells. What we see is the formation of those clusters and surrounded by normal cells. If we measure the typical distance between two cancer cells indicated in blue and normal to normal cells indicated in red, this distribution, you see at the very beginning, they are homogeneously mixed. So we have a good mix, so everything, and they are sparse, there are not so many cells within the dish, so everything is fine. The typical distances are pretty much similar. However, as time goes, we have the cancer cells, they start to get tight, closer to each other, and comparatively, the normal cells, they are more further apart. And by the end, when we reach confluence, what we see is that the cancer cells are typically very close to each other in comparison to normal cells that can be further apart. This region here is going to be explained on the next slide. As you can see, it is predicting that, how long do I have? 5 things. If you take a look, there is this, the peak is not well concentrated around the mode, but it has a big amount of cells that are in a closer distance comparatively to, even comparable to the mode of the tumor cells. So what we saw is the following. If we look carefully, carelessly here, we may think, oh, all the typical distances are pretty much similar. But if we take a look at the interface between normal and tumor cells, what we see is the following. They are closer to each other than the normal cells that are further away from the cluster of tumor cells. So what we see is that we measured, we designed some squares at the interface between the normal and tumor cells and measured the typical distance between those cells in that region. And what we see is that the normal cells that are nearby the interface with the tumor cells, they are closer to each other than the ones that are further away. So what this indicates for us is that tumor cells not only are capable of getting closer to each other, but somehow there is some signaling here this is a question for experimentalists. There is some signaling here that is telling those cells that they should be tighter. A question is this signaling sent by tumor cells, or is this signaling sent by normal cells to say, oh, attack those tumor cells because they are trouble for us? So this is a question for experimentalists. If there's someone here, please help us. So those are further results. What we did is, at the first simulations, what we had is that all the rates, the cell division rate for cancer and normal cells were the same, death rates were the same, and so on. Here what we did is, let's consider that the normal cells division rate becomes bigger than the tumor cells division rate. And then this is the final density of tumor and normal cells. And what we see is that when the normal cells division rate is about 6 times greater than the tumor cells, the normal cells may become prevalent within the domain at the steady state regime by this region, letter D. If we have this condition here, of course, we have a prevalence of tumor cells, and here we have some balance between the two cell populations, okay? And this is the time dynamics of those two. And here, in order to get a further comparison between our simulations and the experimental data, what we did is, okay, let us analyze the clusters. So we analyze the clusters of tumor cells, evaluating their area from experiments and simulations, the perimeter of the clusters from experiments and simulations, convex hull, and the ratio between major and minor axes of our clusters. And what we got is a very, especially if we just consider the modes, a very good relationship between the two, both simulation and experimental data. So just to give some more perspectives, now that we got a model that seems to be working well for experiments considering 2 species, we would like to extend it for 3 species. So instead of getting melanoma and keratinocyte, what we would like to get is a picture where we have keratinocyte and melanocyte, and then at some point a melanocyte suffers some mutation, becomes a melanoma cell, and then we see what goes on, because this is much more close. This is closer to what we are seeing in skin cancer, in real skin cancer, when we consider human beings. We have some previous preliminary results. We show that there are some conditions where, depending on the duplication rate of the melanoma, it may not be sufficient for the melanoma to become prevalent in a given tissue. However, if we start growing at some point, it doesn't need to be the biggest division rate. You see it is much smaller than the division rate of both keratinocyte and melanocyte, but it is enough for the melanoma cell to be prevalent. And the only thing that the melanoma cell has that is different from both melanocyte and keratinocyte is that its exclusion diameter is much smaller than the exclusion diameter of both keratinocyte and melanocyte. So it seems that having a smaller exclusion diameter is a sufficient condition for a given cell population to prevail within a given spatial domain. Some further questions that we have is, one of them is how to, using those simulations, how to go from a discrete time towards a continuous time. We have how long do we have? Okay. So some open questions is, this is pretty much, we have a guess for going from discrete to continuous time, still doing simulation. But of course, we would like to get a continuous limit for our approach, consider a system of ODEs. We also would like to know if this approach can be useful for other cancer types or primary cells. We also wonder if a 3D model would be relevant for in vivo experiments. We also wonder if we change the network topology of our grid, would it cause any change in our simulation results? And of course, okay, and this is it. Thank you very much for your attention and to the people I'm working with. Thanks.